Good afternoon, good evening, good morning to everybody. Welcome to this Hadassah webinar, COVID-19, Can We Win the War? Very pleased to have more than 500 people registered for this event from 19 countries, including our esteemed Hadassah International President, Robert Dorfman, calling in from Hong Kong. I'm going to hand over to our moderator, Norman Swan, to introduce our guest panelists, and I wish everybody a most enjoyable and successful webinar.
Thanks, Ron. Shalom Kolam. And may I pay respects to the traditional Aboriginal owners of the lands on which the speakers meet, and in Melbourne, that's the Wurundjeri people, and in Sydney, the Gadigal people, and pay respects to their elders, both past, present, and most important of all, future. For those of you who are not from Australia, it's a fairly recent innovation that we don't welcome to country, but we acknowledge country and acknowledge that we're on Aboriginal land. So thank you all very much for joining, and it's a privilege to be able to be doing this on behalf of Hebrew University and Hadassah Medical Center, both institutions I know well. So here we are. We go back to November. Just a very few months ago, there was only one person in the world, probably in round about November. Could have been. We'll come to Sharon in a minute on that. One person with what will become known as COVID-19, and here we are with millions of people infected. Almost certainly an underestimate. Whatever you're seeing today is an underestimate, and whatever you're seeing in terms of the number of deaths, that's an underestimate as well, for all sorts of reasons which we may get into. So here we are into what is a really awful pandemic, one which some people predicted, and Australia's done quite well so far. There's lots of research going on around the world. So here's an opportunity to touch base with where we are with the pandemic, what we know, what we don't know, what research is going on, both at a fundamental level, at a vaccine level, and at a clinical level. And when we say clinical here, we're talking about beyond test tubes, beyond animals, actually testing treatments on people. On people. And our three guests today are Professor Sharon Lewin. Sharon is an infectious disease physician and an eminent researcher. A lot of her research in the past has been into HIV treatments, to some extent HIV vaccines, and has a very prominent international role in that area, and very respected in infectious diseases, and leads the Doherty Institute, which is Australia's premier infectious disease institute. And in fact, at the Doherty Institute is the National Influenza Center, led by a very eminent virologist, and also has the mathematical modelling group, which has guided policy and planning in the Australian context, and very successfully so. We've got Russell Basser, who's Senior Vice President, Research and Development of Sycoris, which is part of CSL, vaccine developers. Andrew Cuthbertson was going to be coming from CSL, but he unfortunately couldn't make it. And Russell is certainly not second best, first equal. And thanks very much for joining us, and immense experience in both influenza vaccines and the technology of vaccines. And we'll come to that in a minute, because people think the vaccine is only just about the, whether you use the virus, whether you use a bit of the virus, but in fact what you use with the virus to actually induce an immune response is as important. And CSL is a world leader in what's called adjuvants, which is what goes with the virus, what goes with the vaccine to actually make it work. And last and certainly not least, from Jerusalem is Professor Eyal Mishani, who is Director General of the Hadassah Research Fund and also runs the R&D division of Hadassah. So welcome to you all. Sharon, where we're sitting now, you've been following this in detail. Just give us a context for where we are now in terms of this SARS-CoV-2. And we should probably just define our terms, because we assume that it's not a non-expert audience. You know, when we're talking COVID-19, we talk SARS-CoV-2, and just give us a bit of context here.
Yeah, sure. Thank you, Norman, and great pleasure to be part of the webinar. So a bit of nomenclature first. COVID-19 is the name of the disease that the virus causes, and that's characterised largely by fever, cough, pneumonia. 80% of people can clear the infection, have very mild symptoms, and 20% get sick and go to hospital. And on average, 1% die. That's basically the summary statistics from across the world, though that can vary. And the virus that causes the disease is called SARS-CoV-2 or SARS coronavirus 2. I reckon it's a really bad name. It's a big mouthful, but it is basically tells you that it's very similar to the original SARS that had a very limited spread back in 2003, infected 8,000 people and affected far fewer countries. So SARS-CoV-2 or coronavirus 2 is very similar to that original SARS, but it's developed a few really good tricks. And the best trick it's developed is to be spreading when someone is relatively well. And that's, to me, one of the reasons why this has been a virus that has so successfully spread around the world now, with now millions of infections, every country being affected. And if I just could say one statement about the overall pandemic is that every country is in different stages of the pandemic, either because of how they reacted to it or when the virus first arrived on their shores. And there's a lot of very strong similarities between how Australia responded and how Israel responded, and we're the outliers, unfortunately, in what has proven to be a devastating pandemic across the world.
Eyal, before we get to the science here that's going on, just describe what's been happening with the pandemic in Israel. I mean, when you read it from here, Bnei Brak was a no-go area because of the ultra-Orthodox community not following orders, if you like. Presumably Jerusalem, with its high density of ultra-Orthodox, may have had similar problems. Just give us a sense of what's been happening pandemic-wise in Israel.
So now in Israel, there were more than 16,000 people which were infected. Fortunately, the majority of them were already recovered. We have something like 150, 160 patients. Do you hear me?
Yeah, something's gone with the screen, but we'll assume the technical. You're back.
So we have something like 160 people which are still hospitalized in various hospitals in Israel. I think that we are over the first wave, as it looks like. The majority of the patients which are still hospitalized are in mild to moderate condition. We have something like 54 or 53 patients which are in critical conditions. 44 of them are still using the ventilators. And you're right, we saw that the majority of the patients coming from Bnei Brak, Jerusalem, and the other cities which are known to be an ultra-religious communities. And it could be because of many reasons, including the way of living, the culture, and so on. But I think that the bottom line, I think that Israel is over the first wave.
So 16,000 is quite a lot for a country of what, 5 or 6 million. And how many deaths have you had?
We have something like 260. I would like to correct you if it's okay. We are more than 8 million. So when you do the calculation of a patient per 1 million, we are at the lowest level of the table. And it's impressive. I think that the fact that Israel decided to put in quarantine for 2 weeks the people who are coming from Europe, the Far East, and the United States early in March helped us a lot. And another thing we need to remember, people are talking about the R0 or the infection constant, the parameters, how many people one patient will infect. So we need to remember that Israel have a higher temperature. In Israel, for example, we don't have a subway, and all these parameters influence on the infection constant. So I believe that we cannot compare between Israel and north of Italy, or Israel and France and England and New York. But you were right. I think that Israel and Australia have the same environment or similar environment. And that's why we saw that the spreading of the disease was much less than in Northern Europe, in the States and other countries.
I mean, some people have estimated that the reproductive value in New York might have been as high as 6, given the density of the population, whereas in China, when it started, was between 2 and 3. Russell, one of the, I suppose, criticisms of our response earlier on, but also the confusion earlier on, was we probably, like Israel and other developed countries, had developed a pandemic response, but the pandemic response was based on influenza. Mm-hmm. Just give us a sense of what the difference, and people still ask me, what is the difference here between influenza and this disease? Yeah. And also people not following what pandemic influenza is. Just give us a sense of compare and contrast.
So, thanks, Norman. And thanks, Ron, for inviting me. Happy to be able to fill in. So influenza is a completely different class of virus. It mutates a lot more readily than we've now observed coronavirus too. So we end up having to make a new vaccine every year. But probably the biggest difference from getting a vaccine out and being ready is the fact that the way we prepare vaccines varies according to the vaccine. And that's why we've got so many shots on goal with coronavirus, because the traditional way of making influenza vaccine is through growing the virus in either eggs or in chicken eggs or in a bioreactor, and then extracting the virus and purifying it and killing it and purifying it and giving that as a vaccine. And that's not so amenable to different viruses. It doesn't readily, you can prepare certainly a coronavirus vaccine in that way, but you can't readily change the manufacturing to fit in coronavirus. So we're very pandemically prepared for flu because we have the facilities. We make hundreds of millions of doses around the world every year for influenza vaccine. But because coronavirus is a brand new threat, none of us really anticipated, to be truthful, we're not really prepared to manufacture a vaccine at the levels we need. We don't actually know whether a vaccine will work. And there was good evidence from the SARS epidemics, a lot of research that never quite got to completion because SARS died out. So we knew what the target was almost instantly. Once the genetic code was described, it came out of China very quickly. The right sort of vaccine was, or candidate vaccines were prepared, but we actually don't know if it's going to work. So we were quite prepared with flu. We've not been as prepared because we didn't know what this was going to be.
So Sharon, you've got the influenza group there. Just briefly, compare and contrast what you would have expected with influenza, seasonal versus pandemic, and what we got with COVID-19.
Yeah, so there's a few key differences with influenza and coronavirus. As Russell first said, they're different families of viruses. They do cause similar clinical illnesses. Influenza can also be very severe and can also lead to death, but how it leads to death is actually quite different to coronavirus. In influenza, it's usually because you get a secondary bacterial infection and eventually your lungs can't ventilate or transfer oxygen sufficiently. This coronavirus leads to a whole lot of other unusual complications that we haven't seen before, complications like blood clots, like kidney disease. So this coronavirus infects a whole range of other cells beyond the lungs. Another key difference is that everyone has some basic level of immunity to flu. Every year we get a new strain, and your previous experience of responding to flu influences your current response to influenza. So having been exposed multiple times to influenza means that people have some low level of immunity, except when we get a brand new strain of influenza, and that's what's meant by pandemic influenza. In this setting, coronavirus, there's absolutely no prior immunity, even though actually there are these mild coronaviruses that cause coughs and colds. We think that it doesn't give you any protection. And I could go on about the differences, but one other really big difference is what the impact on children. So in flu, children are a major vector for transmitting the virus. Children can also get really sick. The very young and the very old and pregnant women can all get very sick with influenza. With the coronavirus, we haven't quite nailed it with children, but it definitely seems that they don't get very sick. There are these rare complications of severe illness, but they're very rare. Most people don't get sick. And it seems that kids aren't commonly infected either. And in most big population studies that have looked across the population of who got coronavirus, studies in Iceland, studies in China, studies in the US, kids make up about 2% of the total number of infections. That's very different in influenza, where you have a big spike young and a big spike very old. So they're just a few of the things.
And is that because they've had lots of common cold and they've got some background immunity to coronaviruses?
Don't know. It could be a few things. It could be the quality. Actually, there's some really interesting work that's just come out of the Doherty Institute. It's in preprint publication. So just so people know, there's been this flood of literature on coronavirus. I heard a statistic that there's 53,000 papers on coronavirus out there now. And usually when you publish your work, it goes through review by experts and then it gets into a journal. Well, with coronavirus, in order to share information, the information comes out before it gets reviewed. So we have some work showing that the quality, the type of immune response you make when you're a kid and when you're old to coronavirus is actually quite different, and that may determine whether you get sick or not. Also, coronavirus uses this particular receptor that lines our nose and our lungs. It's the same receptor that the original SARS uses, something called ACE2, and that the expression of that receptor may vary with age. It varies with whether you're a smoker or not. It varies whether you've got on anti-blood pressure tablets or not. And that may be a factor of why kids are relatively immune from infection. We don't quite know that. That may give us some good clues for dealing with the virus.
There's a question for you, Sharon, from the audience, which is, just explain the mortality rate. When the Chinese said it was the WHO said it was 3.4%. It's been higher than that in parts of Italy. Some people say it's 0.5%, 1%. Is 1% just a happy average or what?
Yeah, mortality rate is, first of all, it's the number of deaths over the numbers of infections. So measuring deaths is usually pretty accurate. Most countries can count that pretty well, except for the people that die at home, but people that die in hospitals you can count. The denominator is the number of infections. So many of the audience will know about the discussions around testing, and many countries that don't test a lot will have a small denominator and your death rate will seem higher. So let's just say you're testing widely and therefore you're capturing all the people that have mild disease. Your death rate is actually more accurate and it will be lower. So in Australia, I'll just give you an example. We test widely. Our testing rates, I lose track of this, I think it's about 4% of our Victorian population are being tested currently. So they're pretty big numbers. So we know how many infections are out there. And then we can measure deaths on top of infections. In a country that's not testing much at all, so say Indonesia, and they're reporting lots of deaths and fewer infections, you get a distorted death rate. But if I can just summarize my take on what's been reported from China, from the US, from the UK, from Australia, is that overall the death rate is around 1 to 2%. However, when you look into different ages, and that's probably the most important thing, if you look at people over 80, the death rate is about 15 to 20%. Over 70, it's about 6%, and over 60, about 3 to 5%. And then under 60, it's really quite low. So the age distribution is really important. The other thing that's important when you look at country rates of death is the quality of their healthcare. So when a health system gets overwhelmed, like we saw in northern Italy, in New York, in Wuhan, the care is compromised, and so death rates will go up. And the final thing that will affect the numbers is your age distribution in the population. So in Italy, I think, actually, I heard you talk about this, Norman, on your coronavirus. I think it's about 25% of the population are over 65 or something like that. While in China, it's, you know, it's much, much lower. So when you just look at country rates, you can see all the things that will influence how accurately you test, how accurately you count deaths, the age group and the age range of your population, and the quality of your care. All of that will influence it. I think overall it's probably about 1 to 2% across the board.
Eyal, let's come to the research that's going on at Hadassah, and just give us a quick overview of the various studies that are going on, and then I might reverse back to Russell and Sharon about, you know, just some, you know, what's the latest in terms of treatment and vaccine research. But also we've got some questions coming in on testing, including, you know, an antibody test, but we'll come back. I won't ask you about that, Eyal, at this stage, but let's just give us a helicopter view of the research that's going on at Hadassah into SARS-CoV-2 and COVID-19.
So in Hadassah, we are conducting research in 4 levels, I would say. The first one is improving the testing and maybe developing a new testing. And this is for either the PCR or identifying the virus, but also for the antibodies. We are also developing devices to improve protecting the environments, like filters for respirators, etc. And we are working a lot on extracting and making a concentrated solution of antibodies from recovered patients. And we also conducting clinical trials. In the phase of the clinical trials, it's divided into 2 groups of clinical trials. The first one is repurposing known and approved drug and using them to combat the COVID-19. And the second is trying to find a new biomarkers which will serve as a platform for developing new treatments.
So biomarkers, meaning that there might be a sign in you that you're going to be heading for severe disease, and if the blood test lights up or particular finding on X-ray, whatever, then you would be somebody that you would take special care of and might need special treatment.
Correct. For example, you can find in families that the parents were sick, but the children were not sick, and vice versa. So we are trying to find the reason and to see whether we can identify several biomarkers which are responsible for the people who are non-responsive to the virus.
And are any of the studies you're doing, I mean, this is an incredibly short time frame. Normally when you talk about a randomized trial, you're told by the researcher, come back in 5 years and we'll tell you what our findings are. But we're compressing time frames in clinical trials so that you find out answers very, very quickly. So a little bit, are there any preliminary answers, even though they might, as Sharon said, they might not be peer reviewed yet?
So this is very tricky and risky. I don't want to get into too much details about the results, but I can tell you that, for example, we are conducting a research with a compound named camostat mezilat, and Sharon mentioned the gate which the virus goes through inside the cell, which called the ACE2. It's the angiotensin converting enzyme. And what we are trying to do, for example, in one clinical trial is to inhibit the enzyme which escort the virus into the cell through the ACE2. And we started it with 3 patients. What we did, we measured the virus load at the beginning of the treatment and at the end of the treatment, which is 5 days. And we noticed that 4 days post starting the treatment, the viral load was dramatically decreased, and the clinical symptoms were improved significantly. But again, it's only on 3 patients. The problem that we don't have too many patients now in Israel. We don't have too many patients in Hadassah specifically, and we will be very happy to collaborate with another hospital worldwide, either in South America or in the States or in Europe. in order to find out. And there are several other experiments. One of them is with the Avigan, and it's conducted by Professor Ranir Paz, who is a virologist. Avigan is an antiviral compound which target the RNA polymerase. And what we are trying to do is, one, as I mentioned before, to do the repurposing, but also to find the synergism between several drugs, or in other words, to try to use cocktails. For example, one compound which will be antiviral, the other one which will prevent the penetration of the virus into the cell, another one which will prevent the cytokine storm, etc.
The cytokine storm being this overwhelming immune reaction, which Sharon alluded to, which is when the lungs start to fail and probably is the source of the inflammation of the blood vessels and the blood clotting elsewhere in the body, which can be so devastating. Sharon, you've been talking a lot about the idea that probably one drug is not going to be the answer here. It's going to be a cocktail. And that goes back to your experience with HIV.
Yeah, that's right. In HIV, we always use at least 3 drugs. HIV is different though, because it's a chronic viral infection. You can never get rid of it, so it's always there. In coronavirus treatment, the ideal is that you will only treat for, say, 5 to 10 days. And the real challenge with coronavirus treatments is when to treat. So when people actually are not too sick is when they have most of the virus detected in their throat swabs. And that's in the first week. You can easily find virus in your throat swabs, and actually is highest early when you're pretty well. And then over in that second week, the virus is at a lower level, and it's the second and third weeks where people seem to get sick. And at the time when they get really sick and need intensive care, there's actually very little virus found anymore in the throat swab, and it's now a disease of the immune system going a bit crazy. And these treatments are then to calm down your immune system. So I think what's challenging with coronavirus treatments is the timing of when you give the intervention. With HIV, the virus caused all the problems. So you treat the virus at any time, you eliminate the problems on the immune system. And I think in the early studies, especially in China, an extraordinary amount of experimental medicine was being done. Experimental medicine meaning trying all different things in very small studies. And these are often really important studies to give us ideas to do the big studies to find out if something works, because you actually can't tell whether a drug works when you test it in 3, 5, 10, 20 people. The only way you can test a drug really is making a difference is in what we call a randomized study, where we flip a coin and we give half the people the intervention and half the people a dummy drug, and then we can work out whether it's changed. And those randomized studies are, amazingly enough, are only just coming out now, 3 or so months into the epidemic. Anyway, having said all that, yes, most likely we will need combination antivirals. It's a bit hard to throw 2 drugs together before you've tested either one alone. With repurposed drugs, as Eyal was talking about, meaning that we've used these drugs in other settings, we actually have a really good understanding of some of these repurposed drugs, their safety profiles, so it becomes easier to put them together. And I'll just talk about an exciting paper that came out recently from a group in Hong Kong. And Hong Kong has a lot of experience from SARS-1, and their idea they put a cocktail together of 3 drugs that target the virus. They also gave those drugs very early in the onset of symptoms. On average, I think it was within 5 days of symptoms. And they found that the 3 drugs did much better than 1 drug in reducing the amount of virus in the throat swab and also shortening the time to recovery. But that clinical trial was actually done in people with very mild disease, many of whom may have got better on their own, but they had a control arm, and they showed that in the control arm, clearance of the virus was slower.
What were those 3 drugs?
They were interferon beta. So interferon is just a non-specific activator of your immune system and used for many different viruses. The hepatitis viruses, for example, respond well to interferon. ribavirin, another antiviral drug that's active against many viruses, and Kaletra, which is an HIV drug that many people have been testing for activity in COVID-19. And they compared the combination of Kaletra, ribavirin, and interferon to Kaletra alone. And they got this idea because a very similar study was done for SARS back in 2003, and they showed that that cocktail had some benefit.
And coming back to Eyal's point, in countries like Australia, and we run the risk of running out. I mean, it's a good news story, but we run the risk of running out of patients to test these treatments on. I mean, Australia is a pretty useless place to do these studies now because we haven't got anybody.
Exactly, we've got hundreds of ideas and brilliant virologists and things to test. But actually, I think what we need to do, and I'm sure I'll be interested in Eyal's view, is that this is a lesson also for preparedness. Not only preparedness for the next potential wave of infections, which may well come, but preparedness for the future. And so what we've done in Australia is established two major trials. One that targets people in intensive care units, and that's called REMAP-CAP. That's an international study. And one that targets people that are in hospital but not yet in intensive care, and that's called ASCOT, and it's being led out of the Doherty. And we're establishing the capacity to do that study around Australia in 86 sites, so that if there is a second wave, we will be able to use treatments in the context of a clinical trial and answer what is the best drug to use. But at the same time, we're also partnering with countries in Asia, most likely India, but also potentially Singapore, Malaysia, Thailand, where we will be able to complete the trial in collaboration with sites.
I'll come to Eyal in a second, but we've got the inevitable question about hydroxychloroquine. Is it worth it at all?
Yeah. Well, actually, in our trial in ASCOT, we are looking at hydroxychloroquine alone, Kaletra, the HIV drug alone, and in combination, the two of them together. Hydroxychloroquine, it's incredible how many people know about hydroxychloroquine, the powers of the Trump megaphone. Hydroxychloroquine acts against SARS-1 and SARS-CoV-2 in test tube models. It certainly has antiviral activity. It's been now I just looked, about 8 different studies have been presented. In 3 studies, it showed some effect. In 5 studies, it showed no effect. And all of those studies are slightly different, and none of them are perfect, meaning that they're all small. I think 2 out of the 8 studies are randomized. And there's just so much hype and interest in it. And we just haven't done enough yet to know whether it works or not. We still need big randomized studies to answer the question. Would I take hydroxychloroquine to prevent COVID-19? Absolutely no way. There's absolutely no evidence that it works for protection, although there are studies evaluating whether it will protect people from infection.
So Russell, I'd like you to expand a bit on the vaccine challenge from where you sit. I mean, I know that CSL itself is not involved, but you are involved to some extent, I understand, in terms of providing this adjuvant that I talked about, which goes, you've got some really good adjuvant research to actually stimulate the immune system alongside the virus. But from where you sit, just give us a sense of what you know is happening around the place and expand on that challenge. What kind of vaccines? Are we going to see one at all, positive or negative? You know the story.
Yep. So there's about 115 vaccines in development. It's sort of getting back to what I was originally saying, that we need multiple shots on goal, because the average success rate of vaccine development is about maybe 10% if you're lucky. So we might get 9 or 10 successful vaccines, which would hopefully be enough. They're all based on different approaches. The one that's had the most publicity, and there was a bit of a tantalizing bit of evidence that emerged from America this year, was an unproven platform based on the genetic code of the virus, and essentially what this company has, it's called messenger RNA. It's the chemical the body uses to make proteins, and they've made this sort of chemical that injects into the arm as a vaccine, and the body makes the vaccine itself. Very clever, very flexible. They were up and running in 10 days, and the vaccine produces antibodies in about 8 people so far. If you give too much, if you give the dose too high, it's quite toxic. That's 8 people. As Sharon was alluding to, you need a lot of people. And the challenge with this vaccine is going to be, this platform is going to be that whilst it's quick, and there's some really interesting data now, it's never been made in more than a few tens of thousands of doses at any one time, and we need hundreds of millions of doses. So the scaling up of this and any platform of vaccine will be part of the challenge. Probably the most likely approach to work and be manufacturable is something called a subunit vaccine, which is where you make the protein, the target protein that's going to generate the immune response against SARS-CoV-2. You make that protein in a synthetic way in big vats. There's quite a few vaccines that are already being developed. Some of the infrastructure's out there. It just takes a bit longer to convert what we're currently doing into this particular technology. The University of Queensland, with whom we are collaborating, has developed this approach. A number of other companies have something similar. And this is where what you're alluding to, Norman, where we need something to help the immune system recognize that protein, because by itself, it's not particularly powerful in stimulating the immune system. So we add in an extra chemical to boost the immune response. And because you look for an adjuvant that's been in people, and you then take a technology that's proven, whilst it might be a bit slower, it's more likely to succeed in getting vaccine in hundreds of millions of doses into people. And there are a number of, probably 2 or 3 other platforms where there are more than 1 or 2 approaches that are being developed that have similar characteristics to either the RNA, which is very experimental but very flexible and could be fabulous, but never been done before, to something like the recombinant protein that has been done with other vaccines but is a bit slower. Part of the challenge will be, because we're in such a hurry, because there's such a lot of pressure on us to be protected, and vaccines are probably the safest, surest way, if they work, to get what's being referred, what we think of as herd immunity, or sufficient people in the population who have protective immunity that then sort of isolates the virus. It can't jump to someone else because they just reject it. So it sort of hopefully dies out eventually. That's a theory. The challenge is, though, we won't have as much, particularly a lot of data around how safe it is or how efficacious by the time it gets rolled out. People are talking about by the end of the year for emergency use, vaccine being available. It's going to be interesting to see how it plays out, and it's going to be about what is acceptable uncertainty. How much that we don't know about safety, how much that we don't know about efficacy will we accept before the vaccine is given to the community? And it's a really difficult challenge, beyond all the practical things.
So given that normally with a vaccine study, you would immunize, placebo immunize, and then watch these people for a while, for a while, to see in a natural sense whether they get infected or not. And we're coming off the top of the problem with SARS. It was a nice problem, as I said, in 2003, SARS disappeared. Therefore, there was nobody to test the vaccine on. There will be people to test this vaccine on, but where do you stand with human challenge trials, volunteer challenge trials, where people allow themselves to be immunized for the greater good, and immunized and then challenged rather than a long time for natural infection?
Well, I think there's a very acute ethical challenge here.
But is it people go in with their eyes open and they say, I'm prepared to take the risk?
I think that if there's no available treatment and we can't predict with any certainty, unless we can predict with any certainty that a young, otherwise healthy 25-year-old will be able to recover spontaneously from the infection, there is a problem. I think unless we can be that certain, maybe we can identify a group of people for whom a low dose of virus is a challenge that is ethically acceptable. That I think beyond that, Norman, I think the issue is whether it helps us make decisions about a vaccine's efficacy and safety in the high risk and frontline people. And the population we might test in a challenge study may not be truly representative of the risks that the people who we want to give the vaccine to most urgently face with the vaccine. It's a really nice idea. You figure out how to make it safe. You find the people in whom you can do it. You give the vaccine. You give them a dose of virus. Half the people a dose of virus, and the other half of the people didn't get the vaccine. But if it's not translatable, it may not help the whole process.
Eyal, tell us what's going on with vaccine research at Hadassah, if any.
So in Hadassah, what we do, we are not directly involved in vaccine development. I know that the Biological Institution of Israel is working on it. What we do so far, we try to collect blood from a recovered patient. We try to extract the antibodies and make a concentrated solution of antibodies and inject it back into patient. That's what we do in this level, but we are not closely involved in vaccine development.
And that's serum therapy that you're talking about. Are you finding any success with it, or it's in trials?
So this is also in trials. I know that we treated more than 12 patients in Hadassah. I don't know the exact results and how the injection was affected, but I know that the majority of them survive and got over the disease. But this is, I mean, it's in a progress, and we are collaborating with the health ministry in Israel on that, and also with other hospitals in Israel, and also the private sector with a company which make the process to concentrate the solution of the antibody, the hyperimmune gamma.
Somebody was asking Eyal about a breathalyzer or a 60-second breath test that somebody's developed, maybe at Beersheba. I'm not sure. Do you know about that? No.
Not sure.
There's some very practical questions coming through, probably best directed at you, Sharon. One is, what went right in Taiwan that they've only had 400 or 500 cases and an economy that's not locked down?
Yeah, very interesting what happened in Taiwan. Taiwan were one of the first, I think the first country to close its borders with China. And they also went hard and early because they had experience with SARS. So they did extensive testing, contact tracing, isolation, quarantine, all the things that we know work, which we did do in Australia. And they did it very early. And I don't know about social distancing. I suspect you might know, Norman, but they did close their borders with China. There's no doubt for Australia. We closed our borders with China on February the 1st, a day after the Americans did. But we also closed our borders to people coming from China via Thailand or Japan, which is one of the most common routes. So we really stopped migration or seeding of the virus in Australia, which was the major source, and that's what Taiwan did. So their secret to success was closing borders and early decisive action on testing, quarantining, isolation, and I think some social distancing.
I suppose it's kind of related to that. Somebody's asking, how complete is our understanding of how transmission occurs and where it occurs and where the high-risk situations is? So as we in Australia come out of lockdown, and indeed in Israel, as you come out of lockdown, what do we really know? And it's getting quite interesting because some of the intuitive stuff that the public guessed is probably right, that outdoors is not that much of a risk, but indoors is really anything indoors. I'm taking your thunder away. What do we know?
Yeah, no, the evidence is coming, you know, scientific evidence is always what I like to see, not hypothesis. And the evidence, yes, is really quite convincing now about the risk of patching it indoors is about 20 times higher, or is very rare to report outdoors. Certainly, you know, close, the sort of 15 minutes, 1.5 meters apart, that isn't really based on super strong evidence, but is based on some old data. Other risks, there certainly seems that some people are more infectious than other people. It certainly says that some people seem to be more infectious than other people. Temperature, humidity, there's some evidence suggesting more likely to spread with low temperature, high humidity. That's not so clear, I don't think. Then there are, one of the kind of more, we always talk about this being droplet spread. Droplets meaning that when you exhale, there are droplets that land on surfaces, you touch the surface and touch your mouth or eyes, and that's how you get infected. Whether there is some aerosolized spread, meaning that it goes in the air, is also, I think, can happen in certain situations. So we are certainly learning a lot more about how it's transmitted. Peeling away what parts of lockdown really work is one of the most important questions right now, because when Wuhan went into lockdown, it just shut down absolutely everything. We may, we'll now need to learn, as we kind of relax things, we need to learn what were the most effective interventions and whether we really do have to shut down absolutely everything. And I think we showed in Australia we don't. We shut down a lot, but we didn't shut down absolutely everything.
Another question related to that is, why are we so shy about face masks? I mean, for example, in New South Wales, they're saying there's got to be social distancing public transport, yet kids can go back to school, which makes it almost impossible because they're all roads. And the evidence seems to be accumulating that some kind of face mask in public does reduce transmission and probably could avoid the necessity for social distancing now that we've got low levels of virus and we're coming out of it, rather than when we're going in and there was a shortage of face masks. Where do you stand on the evidence for face masks on a fairly mass scale?
Yeah, the issue of masks is really confusing for people. And my understanding of their role has also changed quite a lot. So masks don't protect you from becoming infected. I think that's pretty clear, because when you wear a surgical mask in particular, the surgical mask gets moist in about 10 to 15 minutes, and therefore it no longer blocks viruses coming in. What masks do is they stop a person who is infected from spreading it to others. We've known that for a while, which is why when, say, you have a cough and you think you might be infected and you go along to your GP, we tell you wear a mask before you walk into the surgery, or don't walk into the surgery. So because we know that people can spread or be infectious when they're asymptomatic and no symptoms at all, none of us know whether we're carrying the virus at any time. And therefore masks could potentially work if everyone's wearing them, because you don't know if you're going to be the infected person spreading it to others. And there's really good evidence that if you are infected with a coronavirus, not actually COVID-19, but another coronavirus, and wear a mask, you greatly reduce the amount of virus that's in the immediate vicinity. And whether masks work at a population level as a highly effective intervention and can replace physical distancing, I don't know that we've seen that. There's indirect evidence that countries that have high mask wearing, which is Asia, which is sort of culturally susceptible practice in Asia, even before COVID-19, all of us know when you went to Asia, you saw a lot of people in masks. Well, Japan, Hong Kong, Taiwan, Singapore, they all have very large numbers of people wearing masks and have all had pretty well controlled outbreaks. That to me is what I'd call indirect evidence. It's not enough to say the masks will actually work. The big concern with masks is that they are absolutely needed in healthcare settings. So if we're using them, chomping through them with the public, and it's not having a big effect, you're using up this valuable piece of equipment. Though access to PPE or personal protective equipment at the moment is not a big risk. So where do I come down on masks? I think if you can't I think the policies that are coming out around airlines, not heavily evidence-based, but they make sense to me. If you can't physically distance, then it's a backup way of protection. Whether we need it at scale, not convinced we have the evidence that we need it. And it's a big intervention. It's a major impost on people, cost and inconvenience.
Another question on rapid testing that's had mixed press, whether the rapid tests for PCR, the nose swab and so on, are, some saying they're inaccurate, not worth the money, misleading, and also the confusion between antibody tests and the actual direct test for the virus itself.
Yeah, I'll take you through that. So yeah, when you talk rapid tests, there's two types of rapid tests. There's two different tests, and both of them can have rapid formats. So the most important tests that we currently are working round the clock trying to achieve in Australia are throat swabs. And throat swabs detect the virus, and they detect the virus by looking at the genetic code of the virus. And there are rapid tests for these throat swabs. They're really well validated. They've been used for HIV and tuberculosis for years in remote parts of Australia, as well as in lower middle income countries. And they work really well, and you can get an answer within 30 to 45 minutes. You can't do them at home, but you can do them in what we call not near care tests. You can do them in, in small hospitals or in general practice, and they're very accurate and they're good. The problem at the moment in Australia is you can't access them because everyone around the world wants them, and so we don't have the capability. So we're reliant on doing the throat swab in standard pathology laboratories, but there's lots of plans for better rapid tests and not to use the big manufacturers because basically there's a run on them currently. The other rapid test that has had a lot of press is the blood test. A blood test for a virus, again, is very common. We use it all the time for many viruses, and it doesn't tell you whether you actually have the virus. It tells you whether your body has been exposed in the past to that virus. And in people that have COVID-19, their blood test turns positive usually after 7 to 14 days of infection. So they can't be used to diagnose people that are infectious. They can be used to tell us, oh, you know that cough or cold you had 2 weeks ago, it could have been COVID-19 because your blood test is now positive. So those blood tests can also in a rapid form. Traditionally, you take a blood test, goes to the pathology lab and you get your result in 2 days. Well, you can also have a point of care test for the blood test, which is a bit like a pregnancy test. It's sort of on the spot. The question is how it will be used. I don't think it's that useful clinically to find people that are infectious and need to be isolated. It's very useful in understanding how many people in Australia have been infected. That's a bit challenging because we've got very low rates of infection. And if a positive blood test means that you're immune from getting reinfected, and we don't know the answer to that, but if it does mean that, and it's highly likely it does, it may tell you that you're immune and you don't need the vaccine or you don't need to protect yourself. So we still try to work that one out.
Eyal, tell me how important you think international collaboration is here.
You know, when we started to work on research on COVID-19, we didn't have enough time to establish collaboration, international collaboration. So we started it with the other hospitals and the academic institutions like Weizmann Institution and Tel Aviv University, and of course the Hebrew University. But we are trying now to create collaboration with the States and with Europe, and we believe in collaboration on a daily basis. We have a collaboration on other projects with Europe and the United States. So I think that, you know, when you come up with something, for example, we developed together with the Hebrew University a way to purify the genetic material of the virus on magnetic beads, and we believe that we can shorten the time of the PCR from 4 hours, the whole process, into something like 2 hours. We also developed a method to flag the genetic material of each sample coming from a specific patient, and by that we can increase the amount of samples that we test by the pooling method and sequencer. So again, once you do it and you would like to validate it and you would like to test it, we need to have an international collaboration to test it in other countries, to test it in countries where they have, unfortunately, more patients and more samples to test. So I believe that one, sharing the knowledge and the experience, and I prefer that it will be done, as Sharon said, through peer review in established journal. Second is to try to enrich each other with new ideas, and there are hundreds of ideas every day they're flowing into the hospital. And three is to create a multi-center clinical trials that will be validated and that we will be able to learn something from it.
Thank you very much, Eyal. Can I thank Russell as well for joining us from Cyprus, and also to Professor Sharon Lewin from the Doherty Institute. I've learned a lot. It's been a fantastic hour. I hope you all agree, the large audience that's joined us, and I'll hand back to Ron.
Thank you, Norman. Thank you, Sharon, Russell, and Eyal. I agree with you wholeheartedly, Norman. I think it's been a very rewarding hour and has added a great deal. And from this Hadassah Corona cast, I'm not, I didn't ask for permission to use the copyright on the name, but everybody should know that Norman has been a wonderful advocate for knowledge over the last 3 months with his daily insights into what's happening with the coronavirus. And we thank him, as Australians, we thank him for the work that he's doing. And I think on behalf of Hadassah, Hadassah Australia Medical Research and Collaboration Foundation, Hadassah Hospital, to all of the participants who have been with us, the webinar will be available on video and on YouTube very shortly. Thank you one and all, and good night and good evening and good day.
Thank you.